Hello, I'm Suzanne Roussan, and in this experiment I'm going to be studying the longevity gene daf-16 as it's expressed in the mutant C. elegans, the mutants CB1370 and DR1565, as compared to its N2 wild type. So C. elegans are small, transparent nematodes. They live about 23 days in their wild type form. Over 80% of their genes have a human ortholog, and many of them are mammalian homologs. So they make a great species to study for a broad application. Within the C. elegans species are several mutant strains, for instance the CB and the DR like I mentioned, and some of them are very long-lived in comparison to their wild-type counterparts, up to 57 days. You can see here this graph. It shows the wild type, which lives a mean 20 days, and the daf-2 mutants live a mean 43 days. But they can also live up to 57 days. So one of the main factors regulating this lifespan is the insulin-like growth factor, IGF-1, the signaling pathway, otherwise known as IIS. So as you can see here, the mutants with the daf-2 genome receives the insulin-like peptides, and it comes through into the cell and alters the daf-16 or activates it, and the daf-16 is one of three components that's responsible for stress response and longevity. In short, this is saying if we compare the gene expression of daf-16 in these long-lived mutants and compare that to the daf-16 gene expression into the N2 wild types, this might give us a further look into the mechanisms that causes the longevity in these mutants. So the methods, the overall steps that I used in this experiment, first I used the N2 wild type as the control strain, and then to select relevant daf-2 mutant strains of the C. elegans, CB1370 and DR1565. Then we selected the reference genes for the normalization against daf-16, PMP3 and CDC42. Then the protocols begin with isolating the RNA using reverse transcriptase to take that RNA isolated and generate cDNA, including a no RT control for each of the strains. I then created serial dilutions at 6 points for the N2 wild type so we can design a standard curve, tagged with fluorophores, and amplified the daf-16 and the reference genes from the cDNA using real-time qPCR, including a no template control, of course. Using the standard curve results gotten from these serial dilutions, I can determine which method to use to analyze the data from the qPCR. And then once that's finished, I can analyze the data to see any difference in gene expression of daf-16 in the long-lived mutants versus the wild-type strain. So first I selected these mutants, the CB1370 and the DR1565, and I did that because they had the daf-2 genotypes. And then the reference genes, I selected the CD42 and I selected the PMP3. So these are the three plates of the different strains of the C. elegans worms. As you can see, there's N2, there's the CB1370 and DR1565. And you notice they do have ample amounts of food, so we could generate enough C. elegans worms for the experiment. And I wanted to note here that a longer centrifuge time was necessary from previous experience in order to pellet the material from the worms, because the pellets were very fragile and they easily redissolved back into the solution. So it was important to centrifuge for 2 minutes instead of the original 30 seconds. And then it was also important to carefully pipette out the supernatant, because from previous experience, when I went to decant the supernatant out, the pellet came with it. So instead I pipetted out the supernatant and just left the pellet in the bottom of the collection tube. After that, I lysed the cells to extract the RNA by adding the lysis solution, and then took the pellet, with lysis solution in it through a series of what's called cracking them open by alternating flash freezing them with dry ice and heating them with an 85°C heat block. And I did this 3 times. This opens the cells and extracts the RNA more easily. Then I added the nuclease-free water and proteinase K mixed thoroughly by pipetting because I did not want to disturb the RNA by vortexing. And then I incubated at 55°C for 10 minutes so the enzyme, the proteinase K, could remove the endogenous nucleases in the samples. Then the next step was to clear out the debris. And I did that by centrifuging the sample through the blue filtration column and kept the flow through because the RNA is in the flow through. I then added ethanol to the flow through and mixed thoroughly. Then I transferred this to the red binding column and centrifuged the lysate through the binding column. The RNA ends up sticking to the binding column, and so the rest of the flow through is just to be discarded. It's all the little bits left that's not RNA that was digested up through the previous steps. Then I used DNase to clean out the remaining genomic DNA. The DNase is an enzyme that will chew up the genomic DNA and leave the RNA alone. I pipetted it instead of vortexing because it's very important not to disturb the DNase because I could deactivate the enzyme. Then I centrifuged one more time to just dry out the column. This leaves the RNA attached to the red binding column. Then I used the elution solution, which breaks the RNA free of the binding column, and I centrifuged this so the RNA ends up in the elution solution and is collected at the bottom of the collection tube. Now, I want to point out that due to previous attempts resulting in little to no RNA, I used the same elution solution that was centrifuged through. I collected the flow through by pipetting it up out of the collection tube, and then I put it back into the binding column again and then centrifuged it again. Then I collected that flow through. So we ran it through the filter twice in order to get the maximum amount of RNA. The next protocol is generating the cDNA from that RNA collected, and each RNA sample was aliquoted to be standardized of about 830 nanograms in each reaction. I calculated this from doing a nanodrop of the RNA extracted and used those concentration readings to standardize this quantity of RNA. 3 samples, one of each strain, were set aside for the no RT controls. So that leaves 3 reactions, each per strain, for the reverse transcriptase. I then used 4 microliters of 5X iScript reverse transcription supermix for each reaction, added the aliquoted amounts of RNA, and nuclease-free water. I should mention here that all water used throughout this entire experiment was nuclease-free because we don't want any added nucleases in the solution, in the mixture, because that could possibly be amplified and we would get wrong readings. We brought each reaction to a total of 20 microliters each, and we also took these 3 no RT controls, 1 from each strain, and put them in a separate iScript supermix. This time it was the iScript no RT supermix. Water was added to bring each reaction to a total of 20 microliters. I placed all the samples in the PCR thermocycler for reverse transcription. Priming was done for the first 5 minutes at 25°C. Reverse transcription took place at 46°C for 20 minutes. And then reverse transcriptase inactivation needed to occur, and that's at a higher temperature at 95°C for 1 minute. And then the thermocycler was set at a 4°C hold afterwards. I collected the samples shortly after the cycle was finished. After the reverse transcriptase, the cDNA samples generated for each strain were recombined into one tube, because before I had made 3 samples each for the reverse transcriptase reaction, just because the reverse transcriptase would be more effective if they were divided into smaller samples. So now I recombined those reverse transcriptase samples into one tube, making one large sample of cDNA each strain of C. elegans. The no reverse transcriptase cDNA samples were kept separately, of course. I then made serial dilutions of the wild type into control strain, and I also did a 110 dilution for each mutant strain, and this is all getting ready for the qPCR reaction. So I made a 6-point serial dilutions at 110, 120, 140, 160, 180, and 100. This is all, I should add, done on ice to maintain the cDNA integrity. And these were all done in triplicates also. The serial dilutions were done in triplicates, and the mutant samples were done in triplicates and diluted to 110. This is a list of the reactions prepared. Triplicates of all serial dilutions of N2 for daf-16 and PMP3. Meanwhile, Crystal Camp was doing the triplicates of all serial dilutions for daf-16 and the reference gene CDC42. I did triplicates of a 110 dilution of CB for daf-16 and PMP, and we did triplicates of a 110 for daf-16 and CDC42. Triplicates for 110 for DR, triplicates for 110 for DR, each for separate reference genes. One no RT control each of the N2, CB, and DR for daf-16 and PMP3, and also for daf-16 and CDC42. And then one no template control for the daf-16 and PMP3, one no template control for the daf-16 and CDC42. So here is the list of the master mix that was made. This is using the JS-TAC Ready Mix. So we use JS-TAC Ready Mix, the gene of interest, which is the daf-16, the forward and reverse primers and the probe, also the reference primers and the probe, and it should be noted that multiplex reactions were done in sets of 2 because there was the gene of interest, which was the daf-16, and one reference gene, which was the PMP, and then the other sets of samples made was the gene of interest and the CDC reference gene. Additional magnesium chloride was added to support the multiplex reactions, and the water was added to bring the total volume to 23 microliters per reaction. 23 microliters because then the actual cDNA was put in a volume of 2 microliters into each reaction, and this was put into a PCR 96 well tray. The contents were briefly centrifuged just to collect all the liquid to the bottom of the tubes, and I used the Strategene MX3005 qPCR machine. The first stage was for the hot start PCR. It was 94 degrees Celsius for 2 minutes, and then 40 cycles of alternating 94 degrees Celsius for 15 seconds and 60 degrees Celsius for 60 seconds. Now, when it comes to the data analysis, this is when the standard curves are used. I take the N2 dilutions and create a standard curve . And by looking at the standard curves, I adjusted the standard curves so the R2 number was as close to 100% as I could get. It was around 99%. And by looking at calculating the efficiency of these adjusted standard curves, I could decide what form of data analysis to use, whether or not I wanted to use the delta-delta CT method, which if the efficiencies had less of a 10% difference, or if the efficiencies had over a 10% difference, and this is comparing the FAM dialane which is the gene of interest, versus the hexadiolane, which is the reference gene. If their efficiencies were over a 10% difference, then I used the efficiency correction method. Also, I should add that the no template control and the no RT controls were also evaluated afterwards for unwanted primer interaction and contamination of genomic DNA, anything that would affect the results, the amplification results of the qPCR. So from the result from the RNA extraction, the very first step, here are nanodrop results. This is N2, and this is CB, and this is DR. And as you can see, there's already a problem with salt contamination. As you can see, this dip up in the upper left-hand corner. DR, not so much. But for the N2 and the CB, there was salt contamination. However, there was enough concentration of nanograms per microliter of each of the RNA samples of each of the strains to continue with the experiment. For the reverse transcription, I standardized the amount of RNA in each of the reverse transcription samples to be around 832. I used different volumes of the RNA extractions in order to get to that level. And I made 4 reactions of each out of the 50 microliters of each sample of cDNA that was generated. Here are the serial dilutions that I made and the procedures that I used. Starting with 1 unit as a baseline, I put 10% of the total volume of cDNA into the collection tube and 90% of water, which gives me a 110 dilution. And this would give me an 11.09 nanograms per microliter concentration. I then took that 110 dilution and did a 50-50 ratio to give a 120 dilution, which gave me a 5.545 nanograms per microliter concentration, and so on and so forth, doing serial dilutions of 140, 160, 180, and 1/100. So the results of the standard curves, these were the altered standard curves because I altered for R2 as close to 100% as possible. It was around 99%. So for the gene of interest, daf-16, my adjusted standard curve had an R2 of 99.84%, and the slope was negative 5.2751, which gave us an efficiency of 54.73%. The PMP reference gene standard curve was adjusted for 99.4% for the R2 and a slope of negative 4.2868, which gave us an efficiency of 71.11%. The daf-16 with the other reference gene, the CDC42, was also repeated in serial dilution, this being adjusted for an R2 value of 99.48% and a slope of negative 2.7717 gave us an efficiency for the gene of interest at 129.50%. The dilution standard curve for the reference gene, CDC42, was again altered to get a better R2 value, and the slope was negative 2.6628, which gave us an efficiency of 137.43%. So for the percent expression, for the daf-16 gene of interest and PMP reference gene, I had to use the corrected for efficiency calculation because the efficiencies were more different than 10% between the gene of interest and the reference gene. So in the CB mutant, the daf-16 gene was expressed at 136.26% in comparison to the wild type N2, and the daf-16 gene in the DR mutant was not expressed as much as the N2 wild type, only at 73.18. Now, in the comparison of daf-16 with the CDC42 normalizing reference gene, gave complete opposite results. The CB and the N2 wild type were very, very similar. They were around 100. However, the DR was expressed at over 140. So as you can see, these two results were very contrary to each other. Now, looking at the negative controls to find out if this will give us any indication as to why those results were so contrary. As you can see, the negative controls did have some amplification and in the daf-16 and PMP3, and they also had an early spike amplification in the daf-16 and CDC42. So here is the shoulder of the no template controls that we were just looking at. And as you can see, the daf-16 line actually went over the threshold, and the PMP3 also created a shoulder, but it was under the threshold. And also in the other samples with the other reference gene, there were spikes of the daf-16, a very tall spike and a very short spike in the same cycle with the CDC42, but these were both beneath the threshold. The no RT controls for my daf-16 and PMP3 went as anticipated. There was no amplification. However, in the daf-16 and CDC42, in the same place that the no template control spiked, there was amplification of this no RT control spiking. So to sum up, the mutant strains selected were specific to the daf-2 genome and not specifically to daf-16 activity. The RNA extracted showed salt contamination, but it was enough concentration of RNA to proceed. The efficiencies from the adjusted standard curves were low for DAF and PMP and high for DAF and CNC. The percent expression rate when normalized to the reference genes were very contrary to each other. So the DAF PMP showed higher expression of daf-16 in CB than the wild type N2 and lower in DR, whereas the daf-CDC showed similar daf-16 expression in CB and the wild type, but higher in DR. When looking at the negative controls, the no template control multiplex reaction indicated primer interactions for both of the reference genes due to those shoulders mentioned. daf-PMP showed an early amplification shoulder crossing the threshold, and the daf-CDC showed an early spike below the threshold. The no RT controls for daf-PMP showed no amplification, so those results went well. However, the no RT control for the daf-CDC showed early CT spikes indicating heavy leftover genomic DNA from the RNA extraction. So the conclusion to this is that I can't really come to a conclusion. The results from the percent expression are inconclusive because of the contrary readings, because the CB was very high in one and very low in the other one. The results conflicted with each other. These readings could possibly be due to the primer interactions or primer dimers, as we saw in the no template control, or other interactions within the multiplex reaction, which would either inhibit or inflate the amplification readings of the gene of interest or the reference genes. So my suggested continuation of the experiments could contain one or more of these possible adjustments. One, the primer optimization, or investigating possible redesign of the primers so they don't interact. I'd suggest a repeat of the experiment using singleplex reactions to rule out any multiplex interactions that may have inhibited or inflated the numbers. New RNA isolation doing another nanodrop reading to make sure that this time the contamination is absent. And the mutants selected should be possibly known for their daf-16 activity and not just the general daf2 genome, because it's possible that the daf-16 activity was not expressed correctly. Just because they are daf2 genome, it does not mean that the daf-16 gene within those mutants was being expressed. I would also suggest use 3 reference genes for endogenous control instead of the 2, and select reference genes specifically without known interactions with the other elements contained in the reaction, such as the primers or the daf-16. And finally, and I think this is an interesting point to make, I think the C. elegans used, both the wild type and the mutant strains, should be raised in a diet-specific conditions, controlling the insulin production. Since the daf-16 activity is completely affected by the IIS signaling pathway, controlling the diet under specific conditions and limiting the caloric intake could minimize any variables in the IIS pathway, which would eliminate any variables that could have been affecting the daf-16 activity. Referring back to the picture showing the petri dishes with the C. elegans in them, they were definitely fed a lot, and if they were fed possibly less, if there was enough time and resources to raise them on a restricted diet, this could control the insulin production, which would eliminate any variables in the IIS pathway affecting the daf-16 activity and keep them all even so they can be more appropriately compared. So these are just some of the references that I used for the background research. Feel free to check up on them. They're very interesting papers. Thank you.